Hey everyone, this is week 7. I can't believe we're already almost done with this course. But anyhow, so you're probably wondering what is left. What are the next steps in these final 2 weeks, week 7 and week 8? So what you have left is quiz 3 and a poster presentation. We're kind of doing like a mini mock conference, even though we're not going anywhere. What I'll do is put everybody's posters together in one PowerPoint and send it around so you can see, all 15 posters or 13 posters. So that will be kind of serve like our mini online virtual conference. But again, so I originally had them do Sunday night, but because of the holiday and then posting this on Wednesday, I wanted to give you just additional time to make sure you had enough time. So again, quiz 3 and the poster presentation, or the poster PowerPoint slide, is now due December 10th. You also have your final paper, and that's going to be due Saturday, December 14th, and that's because that's the official last day of classes. However, if you submit Sunday night, I'm not going to penalize you. So you have an extension per se, but I just kind of have to list, it being due on the last day of class. But again, if you submit Sunday night, I won't be penalizing, but I do just have to turn around the grades for this class, and from the first part of fall, I have to get those grades in fairly quickly. Also, just as a reminder, I posted examples of final grant submissions from previous courses, and I added an additional one in the folder that you have this week, just so you can see one example from me, but then also 3 different student examples. And again, there's one slight difference in that I had them put their pilot data in the data analysis section. But technically that's not right. So starting from this class and having you put your mini intervention results in the approach section, which should have a subsection called pilot study or formative work. So that's just one example and one difference from the examples that I'm showing you online. But definitely everything else about their paper should be fairly simple as far as what I'm looking for. Okay, so really what I'm going to go over right now is just a little bit, getting right into the writing lab. It's the data analysis part, looking at your data, and then a little bit of reporting. But a lot of this we've covered in PH 504, so you've done data analysis in that class. And then I also did a lecture in 507 on data analysis. So I put a link to that where I really kind of get into bivariate analyses, so you can check out that link also within our week 7 module for this current course. But so most of what I'm going to go over today is mostly about your writing lab and your poster and the things that you need to do for week 7. Okay, so again, our class project is a paper, but then also a poster. So I'm just kind of showing you, what is in the syllabus. And so everyone should have already done writing lab on specific games. We did a writing lab on innovation and approach with pilot study results. So this is your results from your mini intervention. And then data analysis with table shells. So you want to at least have some table shells, even if you don't have any data in them. And again, it's just a way to kind of practice showing your data. So data analysis was last week and this week. And it's different for everyone. Everyone had different topics. So I do have as one of your to-dos to schedule a meeting with me if you want to go over your survey, the data, your paper. I made some additional available times just for that. So just look out for that link. And then you want to think about your implications, strengths, and weaknesses. Your strengths and weaknesses are always about your study design and the study itself. So think about it in those terms. And the implications, you're kind of broadening, if you are able to accomplish the research that you're proposing, what are the logical next steps? What is it? What are the implications, public health implications? What impact are you making? That's kind of where the types of things you might add into the implications. And then for your appendices, your lesson plan, really that's just the PowerPoint that you already did, which was considered your mini intervention. Print out, all of them are online, but if you can have a paper copy of the survey, that way I can see it. So you're going to add all of these to your appendices. Of course, have your budget and budget justification, which we did, writing lab another week. And then have your SPS output, even if it's just for descriptive statistics. I realized some people were not able to export into Excel, and therefore you couldn't do SPSS. But what you could do is use my sample data set that I use. I just created one really quickly. So you can do that and just do some really simple descriptives and bivariate analyses. Or you can print out from your site, whatever you used for your online survey, you can print out some of those descriptives. But I just want to see a little bit of, that you looked at the data and what that might have looked like. But especially if you're able to open your own data in SPSS, that's even better. But if not, feel free to use the mini data set that I uploaded into our week 7 module as well. It's just super simple, but it allows you to get some practice if you weren't able to export your data from whichever platform that you used. And then just a reminder, the intervention, we already did that. Poster presentation, again, due December 10th. And then lastly is your final paper. So this is basically the culminating projects for this course. Okay, so some very, very basic stats review. And again, I have a link to my other lecture from 507 where I go more into detail about how to think about some of these basic stats, but I'm just going to review it really quickly in the next couple minutes. The main thing that you really want to have a good grasp on, especially when you're working with data of any kind, is just to know what type of variables you have. Is it nominal, some type of categorization? Is it ordinal, kind of like Likert scales, very likely to agree or not likely? Or is it interval data or ratio? So again, most will be kind of ratio data because it has a true zero, so zero means something, versus interval data where it doesn't have a true zero, kind of like temperature. Zero temperature, there's still a temperature. There's even negative temperature. But true ratio data, a true zero means zero actually means zero zip, there's nothing there. So if you have zero blood pressure, you have no blood pressure, and you're probably not alive. If you've worked out 0 minutes, you have not worked out at all. So it really means zero is zero, nothing versus interval. So again, just really think about, do you have categorical data, nominal data, which is nominal data? Is it ordinal, like a Likert scale, or is it some type of interval or ratio? Most likely you have ratio data. But just make sure you know what type of variable you have, because that is what makes the difference of understanding what statistic you need to do. But I also want you to think about how you can manipulate the same variable into different types. So let's say that in one of your questions you asked about diastolic blood pressure. And as you can see from this chart, it's ordinal. So you have normal, elevated, hypertension stage 1, stage 2, and emergency care. You should be in the ER or something. So this is ordinal data. But let's say, you're really just interested in normal and elevated versus someone who has high blood pressure. So now you're saying maybe you change the variables to, yes and no, or 1 and 0. Yes, they have high blood pressure, or no, they don't. So in that sense, that would be dichotomous data. So now you've taken ordinal data and you've said, you know what, I'm more interested in yes or no for high blood pressure. And again, that's dichotomous data. But let's say you collected information, hopefully in a way that you could get ratio data, and you had a question that just basically said, what is your diastolic blood pressure? Now that could be from 0 to 120, which would be really bad. But now you have ratio data. So you have a continuous variable, again, from 0 to 120. And that is nice to always collect, one, because it's ratio data, but then you can backtrack if you wanted to then make it dichotomous or then make it ordinal. At least you have the most amount of data. So again, we're looking at the same variable, diastolic blood pressure, but here are 3 different ways you can look at it. You can look at it in an ordinal way, normal, elevated, stage 1, stage 2, emergency. You can look at it in a dichotomous way, so change the variable to yes or no. Or if you did get ratio data, you can look at it as a continuous variable ranging from 0 to 120. So just realize too, you're not always stuck with the information you have, but you do, as much as possible, want to collect data in a way where you're able to already manipulate it. And so, well, one example of this is sometimes people will collect a lot of yes, no answers. Well, if it was, if you wanted to break it apart, eventually like, oh, I wish I would have, had different ranges or had a continuous variable. Well, you can't do that if you only collected yes or no questions with responses. But if you started off collecting the most information you can, so more continuous, then you can decide to break it up however you decide. So those are just some tips for, surveying. But again, just realize and understand what type of variable do you have, but also understand that you can manipulate the variables on the back end to change it, from ordinal to dichotomous, or maybe you have ratio data. So that's just one example of how you can see blood pressure in different variable types. So now that you have a better understanding of what types of variables you have, when you're looking at bivariate analyses, so 2, just remember that you're going to do chi-square if you're looking at a basic 2x2 table. So you have 2 nominal or ordinal variables. But for me, just think about the basic 2x2. So you have time 1, time 2 versus yes, no, or meets, doesn't meet. So you have a basic, again, 2x2 table, and you can do a chi-square because they're both variables are nominal or categorical. You do a t-test if one variable is nominal and the other one is numerical or continuous. So you're basically looking at group differences. But in the case of how you collected your information, you collected time 1, time 2. So that would be your group variable. And then if you had some type of continuous variable like a knowledge scale or age, something that's continuous, then that would be your dependent variable. But since you collected one group twice, you would be looking at dependent t-tests. So again, when you're looking at differences, because you have, time 1, time 2, or yes or no, and you can divide that by looking at a dependent variable that is a ratio or like a continuous variable, then that's when you do a t-test. And again, with our design, it's going to be a dependent t-test because it was one group twice versus 2 different groups of people. And then similar to a t-test, if you had more than 2 groups or differences. Let's say you were looking at race, ethnicity, and you had Black, White, Hispanic, Pacific Islander. There's 4 categories, but you then wanted to look at an average across the categories because you have a numerical or continuous variable. Again, it's like a t-test, it's just more categories, and in that case you would do ANOVA. And then if you had 2 continuous variables, so they're both numerical, they're both most likely ratio data, then you could look at the strength of the relationship by looking at correlations. So again, hopefully this is just a quick reminder of understanding how knowing what type of variables you have will help you decide on which test you should be conducting. And again, there's a link to a lecture that I did on this exact slide and more, I think right there in number 1 in our Cougar course content for this week. So then what to analyze? I guess I put that there just to kind of remind myself to remind you that when you're thinking about the data that you have, again, you're thinking of mostly time 1, time 2, which you can have in a chi-square because that's nominal. It's also appropriate for a t-test if your other variable is continuous because you have time 1, time 2. You might not be doing ANOVA, but also maybe you have 2 continuous variables. In the data set I gave, it's a mixture of continuous and dichotomous. So if you were looking at the continuous, exercise variable and the continuous blood pressure variable, those would be 2 continuous variables that you can kind of see. Is there a relationship between exercise and blood pressure? But again, just really think about what data do you have and what does that tell you about how to analyze it. So hopefully that helps. And if not, feel free to schedule a meeting with me and we can work through your specific data. And sometimes it's a little bit easier to conceptualize. But I did want to point you at least in the right direction if you're using SPSS. Again, hopefully you did this in biostats. So I'm just trying to jog your memory, but when you're doing descriptive statistics, just go to Analyze, Descriptive Statistics. And the first 2, frequencies or descriptives, is how you're going to get your either frequencies or means, standard deviations, ranges, etc. So you're just describing the data. And then if you wanted to do chi-square, you're actually going to click here in crosstabs, and then it's going to pull up this window. And if you think about rows and columns like your 2x2 table, you're going to insert 1 variable for your row and 1 variable for your column. And again, each of those should be nominal or categorical data. And again, for simplicity, you can think of a chi-square for the purposes of this class, like a simple 2x2 table. That would work just fine. And then if you were comparing means, so doing a t-test, again, you're going to go to Analyze, Compare Means, Paired Sample t-test. Again, it's paired because it's the same group twice, it's the same people, so it's considered a dependent t-test. And that's going to pull up this window, and variable 1 and variable 2, you're just going to pull in the 2 variables that are alike, but one is the pre-test and the variable number 2 is whichever variable but the post-test version. So you'll have to make sure that the way you've labeled your data says, for example, blood pressure time 1, blood pressure time 2. And so you'll want to label it that way, and then those would be the 2 variables that you would look at in the paired samples t-test. And then if you had 2 continuous variables, you could do a correlation, and you would click here under Analysis and Correlate, and then what would pop up after that is bivariate correlation, and then you would just select 2 variables that are continuous, and that will give you correlation. So hopefully this was just a good reminder of where to do some of the very basic statistics. And then again, if you need help, I'm always available by Zoom, and I posted again some office hour times that you can sign up for, or schedule a meeting with me at a separate time. Okay, so again, for our writing lab, I want you, last week you explored some of the descriptives, and it's okay if some of you couldn't export all your data. But hopefully you're at least able to look at the descriptives. So what I want to do for this writing lab this week is to, again, look at your data, run some statistics, look at your results. Definitely for everyone, make sure you have at least finished your descriptive statistics. And then when you run the results, make sure you create some data tables that you can include in your mini grant. Again, you can even look at pre-test, post-test just descriptively and say, at pre-test, this is what I found, and at post-test, this is what I found, which a lot of you did in your writing lab last week. And so this week, what I want you to try to do is just experiment with some bivariate analyses. What would make sense to analyze? Are you doing a chi-square, a t-test, some type of correlation? So definitely, practice some of that. I know 504 biostats was a little bit ago, but I definitely wanted to, give you another opportunity to practice some very basic statistics. And sometimes you might even have missing data. So if you happen to be using my sample data, and again, I just created it today, just off the fly, a really small data set. What I did for missing data was I coded it 999. We talked about how to clean data sets last time, and that's kind of running frequencies and making sure everything is in line. But because I added 999 for some of the entries, if the range was 1 to 4, but I have a 999 in there and you ran a range, the range is actually going to be 1 to 999, which is incorrect. And so this is one of the ways that you clean your data set. And you look at it and say, oh, okay, so obviously 999 is going to be coded for missing data. So what you would do in your data view, part of SPSS, is you would look at the column that's labeled missing, click on that, and there's a way for you to indicate 999 equals missing for a specific variable or for all of them. So that way now when you run your frequency, it will say your range is 1 to 4, if that's what it's supposed to be, and you have one missing variable, which would have been the one that was originally coded 999. So just know that even if you have missing data, that's okay. And sometimes in survey coding, we might assign it a certain number, like 999, but you can definitely handle that in SPSS by telling SPSS that for any time you see 999, please code it as missing. And again, you can do that in data view, and there's a specific column called missing. So there's ways to handle missing data. And as I've mentioned a few times already, if for some reason you weren't able to export your data, I still want you to practice and try some of these bivariate analyses. So you're free to use the really small, simple data set that I created, and just use that even in your writing lab or in your report if for some reason you really need data and you couldn't work with the ones that you got. I just want you to know that this task and working with the data and the surveys, it's mostly about learning the process and gaining some of these skills. So what this class is about, at least for this portion of it, is making mistakes. So it's okay if you couldn't export, or it's okay if you ended up asking questions in a way that maybe didn't make sense for your data interpretation. My philosophy is this is really how you learn. And so my goal is that by the end of this class, you'll understand, okay, this is probably how I should have designed a survey, because I realized, what mistakes look like, and how it can hinder, running results. Or maybe I should ask questions, getting as much information as possible as far as ratio data, and not asking yes or no all the time. So this part of the class is really just about the process. So don't worry if you weren't able to export, or if you didn't, if you figured out you didn't design your survey the best way. In week 8, we're going to go over all of this as kind of a review. And again, my goal is more about the skills. Usually after you've taken this class, especially because now you have a little bit of grant writing experience, a lot of my students will, get into a new job, and then I'll get an email saying, I'm so glad you talked a little bit about grant writing, because that's what they're doing now, especially with budgets and budget justification. You typically don't get that in an MPH curriculum. And then also, if you have another job or a new job, and they say, hey, I want you to conduct a survey, hopefully this has given you some insight into how best to create a survey through learning through some of the limitations you might have had. I feel like that's always the best way to remember, is just trying it, making some mistakes, and then improving along the way. So again, don't worry too much if you weren't able to complete an export, or if you didn't have data in a way to do your bivariate analysis. Again, you can use my data set. But for me, this part was more about gaining skills. Anyhow, the last bullet. So again, your writing lab, just really think about your final paper and poster. The poster that I want you to submit by December 10th is really a rough, not a rough draft, but like a summary of your paper. So they really go hand in hand. And again, we've already done writing labs on the specific aims, which could be a header, innovation and approach, which is a big part of your poster. And then you talk about data analysis and implications. So those could be the main headers of your poster. A traditional conference poster is about a specific study. We're kind of doing a poster about a grant. But in any case, I know that this will be the first time for many of you doing a conference style poster. So again, that's the skill that I'm looking for, that you can, look at some of the examples that I posted, use those as templates, and then add the information from your mini grant, so that you're kind of presenting your mini grant in a conference style type poster. And again, just remember to place your pilot study or your mini intervention results in the approach section. So again, that's basically what we're doing for Writing Lab, and then you'll submit your poster by next Tuesday, December 10th. Okay, so again, I did post a very small, simple data set. I think it only has 3 variables. But I just wanted to give you a few clues, because I do want you to explore it and try to interpret it. But MVPA is moderate to vigorous physical activity, and I have the variable for time 1 and time 2 in 2 different ways. So I have it as a continuous variable, which is in minutes. So if it's 0, it's someone didn't exercise at all. If it's 150 minutes, they've exercised that amount of time. As you know, that is what meets weekly requirements. So in the dichotomous version, 1 equals meets, moderate to vigorous physical activity requirements. 0 means it's under 150 minutes. And then I also just added in fake diastolic blood pressure. And again, I have continuous variables, time 1 and time 2. And then I also have a dichotomous one where it's 1 equals high blood pressure and 0 equals normal. And again, this is just diastolic. And then I do have a few missing data, which are 999. So that should help you at least understand the data set a little bit. Again, it's very small and simple. But if you do decide to use this to practice your bivariate analyses, make sure that you label it. So go in there, look for MVPA, continuous time 1. In time 2, sorry, you should label especially the dichotomous one. So go into MVPA dichotomous time 1, and you're going to label 1 equals meets the requirement, 0 equals doesn't meet. And then the same for diastolic for the dichotomous variable. You're going to label it in your data view. 1 equals, high blood pressure, and 0 equals normal. And then, of course, you can also label missing data, which I explained earlier. So here are just some hints about the data set, but again, it's for you to explore and to practice some of the bivariate analyses. Okay, so I've given the data analysis lecture before. Here is a link to the YouTube video. Of course, the link is also up in our week 7 module in Cougar course. So I'm going to end my voiceover here. And please remember to, if you need more help on your survey or interpreting the data or trying to decide what analyses you're going to do, or just questions about the paper or poster in general, I've posted some times that you can sign up for, and if none of those times work, of course, just always feel free to email me and send me some proposed times that you would like to meet. But hopefully, again, my goal for this class is really just to expose you to some of these skills, and hopefully it helps. So let me know how I can support you, and I'll talk to you soon.